Up is Professor Yoel Stuart of Loyola University, who is using eDNA to study life in the Fox River.
Hit them arrows and we'll be good. Thank you.
Hi, everyone. Can you hear me?
Yes.
Okay, thanks. Thanks for being here. Thanks for sticking around for the afternoon session. As introduced, my name is Yoel. I'm a professor. Sorry, what? Can't?
Raise that if you want.
I'll just hold it.
Oh, yeah.
I'm a professor in the biology department at Loyola University. And it's, to echo some of the earlier speakers, it's a pleasure to be here today, talking to this room of great people doing great work. I like to do my acknowledgements first to make sure that there's time and I don't forget. Big thank you to the Friends of the Fox River. They've been gracious enough to allow me to sort of reach out to them and work with them on some of the projects that I've got going. And thanks for the invitation to speak here. I have a number of colleagues who have sort of helped me get started in the work that I'm going to talk about today. Eric Larson and Mark Davis at UIUC INHS are experts in eDNA, and they helped me sort of put this project together. Sophie Peak has been sort of the day-to-day person I've been working with to make the eDNA stuff work. And then the project in the Chicago River I'll talk about today was with Leslie and Phil. And then I've reached the point in my career where I don't actually do any work. All of the work is done by students, like you guys know back there. Couldn't do it without these folks. They spent 6 weeks this past summer in the field, going out to all these sites on the Fox River to collect the water samples that we need for the work that I'll talk to you about. And so I do need to give them a big thank you. So this is a bit out of my field. I'm trained as an evolutionary ecologist. I'm sort of, traditionally been ivory tower basic research. My questions are about how fast is evolution, how repeatable is evolution. I've studied things like anoles in Florida and three-spine stickleback in British Columbia. I've gone to Utah for Daphnia, and I study the big project in the lab right now is fossil stickleback from a 10-million-year-old lake. And so I guess the question is, what am I doing here at the Fox River Summit, where this is a group of people who are boots on the ground, conservation, restoration, management work, doing really good things. And I suppose I'll give the next plug to my funder, which is the Walter Foundation. I wanted to make you all aware of the Walter Foundation. They have a pillar in their, this is a relatively new philanthropy group, and they have a pillar in their sort of foundation. One of their major missions is to restore and conserve biodiversity in the Chicago region and around the world, but they are a place-based group in the Chicago region. They're relatively new. And so I just wanted to make folks aware. And so I've been fortunate to get funding from them. I'm putting roots down in Chicagoland and Illinois, and so I was excited for a chance to do some local research, contribute to the kinds of work that all y'all are doing. So they funded me essentially to build eDNA resources for Illinois fishes. So I'll tell you sort of what eDNA is if you don't already know, and then see if we can figure out how good this tool is. This is motivated by the question, right? This key problem in designing your conservation and designing your management, et cetera, of how many of what species are where and when are they there, right? So we heard the talk this morning by Dave Marshall, right? And he's got all of these lakes that he's trying to find his fish in. We heard the talk about the bumblebees, trying to do all of this work to go find these bumblebees. You might want to know where your endangered species are, your sort of important ecosystem indicators, right? You want to know where your invasive species are, like that milfoil, those hard to catch and cryptic things that are sort of underpin the work that we're all trying to do. Conventional approaches to figuring out that species distribution and abundance. I'll talk about fish in this case, but you can pick your favorite organism, like the work on the bumblebee that was discussed earlier, right? It tends to be labor intensive, right? You got to get out there with the big electric backpacks. You got to get boats that have, electro fishing setups. You got to get a bunch of people out there. You got to drag these gill nets through the environment, right? It's a lot of work. It tends to be expertise intensive, right? You get a big bucket of fish and you got to figure out which fish are there. It's costly, therefore, in time and money. And so this kind of sampling is often temporally limited, right? You can only do it every once in a while. Spatially limited, we can only do it in a few places. And then taxonomically limited, I can only do fish today or fish this year, or I can only do, whatever your focal organism is. So it's hard work. And so maybe there's some other approaches that we can try. This idea of eDNA or environmental DNA is a relatively new idea that's sort of come along as genetic sequencing has gotten much cheaper and the methods have gotten much more straightforward. We are all inputting DNA into the environment all the time, right? So anybody who's a golden retriever owner knows this, right? But it's not just that, it's scales, fur, mucus, pollen, leaves, everything is just out there everywhere. And so we can take advantage of that fact. We can go out into nature, we can collect that DNA. If you're working in aquatic systems like I do, you just stick a pump in the water like this. You filter a bunch of water through that pump, it comes onto this piece of filter paper, and then you take that filter paper, you run it through a bunch of, one clear liquid into the next clear liquid, and magically at the end you have DNA. And so this ends up kind of being like CSI environment. I use this analogy with my students, and they look at me, they're like, what? I'm like, that forensic science show where you can like get fingerprints and stuff, and they're like, oh, that's my parents' favorite show. Time comes for us all. But anyway, so it's kind of like CSI environment, right? And essentially you can sequence whatever you get. And so you can take these things called metabarcoding approaches where you sequence all the DNA that you get back. And if you have a really good database, kind of think about like a fingerprint database, right? If you have a really good database, you can sequence everything. And then you can say, all right, I got this set of A's, C's, and G's. I'm going to call it species A, et cetera, et cetera. You can target more specific groups, right? You can sort of focus your efforts, fishes, mussels, whatever you want. Again, it requires that sort of good database. And then you can even get down to like single species, right? Silver carp, whatever we're interested in. And some studies suggest, right, that once you've got this system set up for whatever your favorite body of water is or whatever, right, you can sort of standardize it. It's relatively cheaper on a kind of per species or per sample basis. The field work is less labor intensive. You could really just have one person going around with a pump, less disruptive to the environment. You're not dragging insane nets through, and less expertise intensive. I am not an ichthyologist. So Dave's talk from this morning, he was showing all those fish. I was like, I don't know what those are. But I can get genetic sequence data and put it back against the database and know what's in that water without having to spend, 20 years of training to learn that kind of stuff. And there's some studies showing that eDNA can be as good or better at detecting species with much less cost and effort. And so that's the case for it. I'll talk a little bit later about why it's not going to be just a perfect panacea or anything, but we'll move on to this. So the title that was put on your program was eDNA from the Fox River. I'm going to bait and switch a little bit here. That data is coming. Okay. So I'm going to give you a case study instead from some work we did on the Chicago River, and then I'll give you some teaser for what we do have from the Fox. So this is based on work in the Chicago River's Wild Mile. Has anybody been to the Wild Mile? There's a few hands up. So this is near Goose Island, on the east side of Goose Island. Urban Rivers has this project going where they're slowly sort of creating this artificial habitat by essentially floating a bunch of habitat, right? So they have these things called floating artificial wetlands, where they create this base and then they plant native plants on top of it, and those plants put their root systems through that floating habitat. It essentially creates this habitat where before there wasn't any. And so here's a picture of the sort of completed wild mile, but you can imagine when this wasn't here in the Chicago River, all you had was this sort of channel, right, with these metal walls, totally straight, no habitat complexity at all. But then by the end of this project, right, they've made these really nice sort of native plant marshes, or at least simulated them. So they've done this amazing work. It's a really sort of beautiful place to walk around. But the question is, what impact is it having? So we used eDNA to try and solve that problem, or try and answer that question. My collaborator, Sophie, back in August of 2024, took 10 filters from within the wild mile. So right here, it's not a mile long yet. They're waiting for funding to extend it. 10 filters from outside the wild mile, a few over here, a few down here. And I'll skip all of the sort of laboratory-based details, but essentially we sequenced that eDNA specifically looking for vertebrates and specifically looking for mussels. And we asked what species were there. This is kind of the summary of what we found. I apologize for folks in the back of the room if you can't see it. But essentially what we found is that this box is species that were shared. So sort of with the yellow header were shared among both the wild mile sites and the sites outside of it, and sort of the regular sort of unrestored channels. And then this column is things that were found only in the restored. And then the last column is the ones that were found only in the sort of channelized portion of the river. And what you can sort of see is that we detected birds, we detected fish, we detected mammals. So we found some invasive things, we found some endangered things. But the number of species is the same in both. But if you sort of do some more stats, and I will walk you through all the stats, so I can do that after this talk if you wish, right? What we did find is that even though the number of species is the same, the sort of distribution and abundance of those species differs, and that there are some species that you'll find more often in one environment versus the other. So things like bluegill, green sunfish, weed shiner, wood duck were significantly associated, found more often in the wild mile, and then things like brown goby, white sucker, creek chub found more often outside of it. And that sort of makes sense. The natural history that, stories you can tell about the species that are found in the wild mile, they tend to prefer that complex structure and vegetation. So it seems like the wild mile that this effort is doing what it's intended to do. We also detected a couple of mussel species in the wild mile and downstream of it. These two species. Urban Rivers was quite excited that we detected this one because they're trying to sort of restock it, reintroduce it into these sites. And then also, I understand from Gary that people were surprised that when the Carpenterville Dam went down, there were a whole bunch of mussels upstream of the dam that nobody knew were there because they hadn't been detected. And maybe eDNA would have done the job if we had been there beforehand. Okay, so this is the Fox River Summit. I'll let you know then what are we doing in the Fox River. So essentially what we want to ask is this approach, that we just used in the Chicago River, is it useful, better, worse, the same, complementary against sort of that conventional sampling approach? So IDNR every 5 years samples the Fox and its tributaries in Illinois. They go to 15 main stem sites and boat electrofish and seine, and then 26 tributary sites in seine, and they'll take every fish that they catch, they'll ID it, they'll weigh it, and they'll return it to the water, although they'll take the small fish and they'll pickle them so they can ID them later. They last sampled in 2022. In June, they did the main stem. In August, they did the tributaries, and they found over 41,000 fish, including some state-threatened species and some non-native species. So we wanted to essentially try and replicate their experimental design. We sampled the same 41 Fox River sites in about 6 weeks, but we also added 20 dam sites, so above and below 10 of the dams on the Fox River, with the understanding that maybe not, they'll get removed. And so we have this baseline data, or we'll have this baseline data to basically say what were these sites like before the dam removal, and maybe we can see how the environments recover. We did some tests of filter size on our eDNA filters to essentially ask if we could get away with not having to use small pore filters that are really slow. We added some temporal replicates to ask if sampling in June gives you a different answer than sampling in July, et cetera, right? So it took us 6 weeks to get all of that, basically almost doubling the amount of sort of sites, et cetera, or time and a half. And we had a field team of 4 to 6 students to give everybody some experience, but it could have been just 2. So just to give you a sense of how much people power you need. And then since we've gotten those samples, I've had a technician working at about 50% time to process them all. This is the first time we're doing this in my lab, so things have gone fairly slow, et cetera, but I think it'll be faster next time. And so just to get back to my point from earlier, right, I think I view these techniques as complementary, right? The sort of conventional approach and then this eDNA approach. Conventional approaches are well established. They sort of plug easily into things like the IBI, which is a measure of water quality. It's really valuable to have the fish in hand, and you can much better estimate abundance. Right? But then it's the cons that we've talked about, including that sort of expertise bit, like here's a central stone roller and there's a large scale stone roller. And I guess we can squint and see differences, but like that takes a lot of effort. Pros of the eDNA approach, I've sort of mentioned, right? Once you've got it going, it can be fairly fast. You can outsource some of the stuff. The big critique is, does the number of sequences that you get equal abundance? So if I get a filter that has a lot of species X on it, is that because there was a dead species, individual of species X right next to my filter, right? And there's ways around sort of dealing with that problem, but that's an open question in the field. So I think these two methods will work together. I think the experts who have that natural history knowledge should not, that knowledge shouldn't just sort of be ignored at the altar of genetic sequencing, right? I think that's highly valuable. And so I think these are going to be complementary. And so if y'all invite me back next year, I'm happy to sort of report how this eDNA approach does. Maybe it'll be better, maybe it'll be worse, but we're processing all the samples and doing all the analysis right now. What I will show is this slide. So this is a student independent project by my student Ebony, who went to the same 5 sites, once in June, once in October, and did her collections, processed all her samples, and what you have in blue is the species that were detected both in summer and fall, in orange just in the summer, and then in green just in the fall. And so it looks like for the most part, we're detecting a few novel things depending on what time of year we sample. And I'll leave it there. I'll leave this up so you can find your favorite fish species up there, and I'm happy to take questions while you do that. So, thank you. Or maybe there's not time. Stop.
Good question. We need to get our next speakers up.
I think go ahead and answer those.
Yes. So the question was, does the DNA only detect the species, or does it give you information on the health? You're not going to be doing sort of, probably not going to be detecting like recessive disease variation in your population using eDNA. It's going to be presence and hopefully abundance as well.